Okay, let's review homework and study guide package. So basically, know the organs of alimentary canal, pretty much the anatomy question, like, liver is not a part, small intestine is the part. Primary digestive, the ones that comprise alimentary canal, secondary that do not. So primary, you get the gist, and secondary should be like liver and pancreas. So that's the difference between primary and secondary digestive. Primary comprise alimentary canal. Visceral peritoneum covers intraperitoneal organs. Parietal peritoneum covers peritoneal cavity. It's the serous membrane. Posterior to peritoneal cavity, retroperitoneal, mesentery, immune responses, mostly, and blood supply, and connecting organs and holding them together. Main digestive activities, so they kind of listed here. I don't remember which slide that is, but like you should be able, mechanical activities, chemical activities, that kind of stuff. Okay, so like segmentation and peristalsis and mastication and deglutition and defecation and churning are all mechanical digestion. Secretion and absorption, chemical digestive activities. External stimuli, digestive activities, sight, smell, taste of food. Virtually all of them act upon the sensory part, and that's going to be external stimuli will act on somatic as well as autonomic, but we can say, taste and smell and sight, they all act upon somatic. Okay, now what's important to understand here, the digestive reflexes, all of them are autonomic. Okay, and effectors are smooth muscles and various kinds of exocrine glands. Endpoint effects is digestion. Okay, muscles move the food around and glands secrete chemicals that digest food. Internal stimuli for digestive tract: stretch of digestive organs and change in the pH or chemical composition of the food while in the digestive tract. So receptors, it will be chemoreceptors and mechanoreceptors. Stimuli, so stretch acts on the mechanoreceptors, while chemicals act on the chemoreceptors. And sensory inputs go first and foremost into the medulla oblongata. So you can basically think of it this way. Practically all digestive reflexes mediated by medulla oblongata. Gut-brain, it's the, I'm trying to fix it. Okay, doesn't work for some reason. Gut-brain is okay. Gut-brain is in the wall of the stomach or small intestine or large intestine, basically a nerve plexus. Folares, no folares. Mucosa, submucosa, muscularis externa, and serosa. No substructures. I'm not going to go over these guys. Like in mucosal layer, there's epithelium, lamina propria, that kind of stuff. So just know the functions of those. Okay. So it kind of explains all of it. Mouth, mastication, chewing, and deglutition, part of it. Two anatomical cavities, buccal cavity and oral cavity. Know the difference between the two. Palate can be soft and hard. The function of uvula is to close the nasopharynx when we swallow. Palatine tonsils carry out the immune function. Another important thing to remember, I'm going to go back for a second to the reflexes. So reflexes are not just autonomic, they are mediated by parasympathetic nervous system. Okay. Intrinsic salivary glands located in the tongue, lips, walls of the oral cavity, and so on, gums, so on and so forth. And they release lingual lipase. Extrinsic salivary glands, please know them, We're talking about submandibular, sublingual, and parotid salivary glands. They produce salivary amylase. Extrinsic gland is the biggest parotid. Submandibular, I think, produces most saliva. Mucin, sticky, water, solution of the water chemicals, lysozyme, antibacterial. Antibacterial, breaks down fat, breaks down carbohydrates. Two types of secretions for saliva, mucus and serous. Salivary glands are activated by parasympathetic nervous system, remember, and it's an autonomic reflex. So parasympathetic activates, sympathetic inhibits, all autonomic reflex. Salivation reflex, food in the mouth, signal to the medulla. Medulla via parasympathetic system stimulates salivary glands to release saliva. Excitation of right, that sympathetic stimulation, salivary secretion will be reduced. If salivary glands would have been surgically removed, people would have troubles digesting food because they wouldn't be able to make a bolus of food. Teeth would rot because there would be no protection against the bacteria, peritonitis, infection of salivary glands. Main complication for males is architis or infertility. The treatment, there's no treatment really. Prevention is vaccine. Main parts of the tooth, know them, the root, the neck, the crown. Layers, enamel, dentin, pulp. In the pulp we have nerves and blood vessels in the pulp cavity. Root canal happens when enamel and dentin rot away and bacteria enter the pulp cavity. Gums are composed of connective tissue and covered by the stratified squamous epithelium. No different types of teeth and they function. It's all in the notes. Know the difference between the deciduous and permanent teeth. Know that premolars present only in permanent. So it's kind of crosses this. Pharynx, anatomically, know where it is. Structures that bound pharynx. Nasal cavity on one side and trachea and esophagus on the other. Larynx and esophagus on the other side. Structures in the head. Pharynx is connected to pharyngotympanic tube, which connects it to the inner ear. Three parts of pharynx: nasopharynx or pharynx laryngopharynx. Remember, nasopharynx, pseudostratified columnar, laryngopharynx, simple stratified squamous. Same goes for oropharynx. Path for the food through the pharynx, mouth, oropharynx, laryngopharynx. Only initial swallowing, moving from mouth into the oropharynx, that's a voluntary step. Swallowing reflex, step by step. Please be able to describe it, such as like food is, moving to oropharynx, stimulation of deglutition center, medulla, then uvula closes your nasopharynx, epiglottis closes larynx, and food goes into the esophagus. So here we got this. So that's pretty much the breakdown of steps of swallowing reflex. Junction between esophagus and stomach, gastroesophageal or lower esophageal sphincter. It controls the movement of food between esophagus and stomach. Inner surface of esophagus, stratified squamous epithelium, so it protects against rubbing forces. Muscularis externa of esophagus, you go skeletal on top, mix of skeletal, smooth in the middle, smooth the bottom. It doesn't have serosa. It has adventitia epithelial cells at the esophagus stomach junction. They change to columnar. So stratified squamous epithelium changes to columnar epithelium. Anatomical parts of the stomach: cardia, fundus, body, and pylorus. Know the function of each. Okay, rugae are folds. Muscularis externa in the stomach has 3 layers. Okay. So not only circular and longitudinal, but also oblique. Other layers that you can find, so pretty much the oblique layer. Inner walls, columnar epithelium, different types of cells such as mucous cells and absorptive cells and G cells and chief cells. Know those types. Okay, parietal cells. Mucous cells, mucus cells produce mucus that protects stomach. Stem cells regenerate all other cells in the stomach epithelium. Parietal cells, hydrochloric acid, they absorb vitamin B12. Chief cells produce pepsinogen. Mucous neck cells, unclear. Enteroendocrine and G cells produce hormones. G cells, specifically gastrin. Enteroendocrine cells, for instance, produce histamine, among others. Epithelial composition between body and fundus, cardi and pylorus. Remember, body and fundus are two parts where most digestion occur, so they have a lot of parietal and chief cells. Pylorus has a lot of G cells because it regulates the activity through gastrin. Main components of secretions, we kind of discussed it. Increases gastrin secretion. I have it in the lecture, so basically gastrin secretion is increased when food is in the stomach. So when pH increases or stomach gets stretched, G cells secrete gastrin. Main producer of gastrin is pylorus. Gastrin secretion on the stomach. Gastrin directly stimulates parietal cells to release more hydrochloric acid. And it increases motility. It increases churning in the stomach. Small intestine, so motility of small intestine increases under gastrin. Ileocecal valve opens. Large intestinal motility increases. Ghrelin is the hormone of hunger, so when you're hungry, when you have an empty stomach, ghrelin secretion goes up, effects slowing down digestion. Serotonin, presence of food, effect on the stomach, increased smooth muscle activity. Release of histamine is stimulated by food. Cells, ECL, enterochromaffin-like cells release histamine, and now that's kind of a weird, so it's actually typo here. So histamine directly stimulates gastrin. Okay, so histamine stimulates gastrin, and cells that are target for histamine are G cells. Benadryl would slow down digestive process, resulting in pretty much constipation. Parietal cells produce hydrochloric acid. CO2 interacts with water. Carbonic acid is formed. It's dissociated to hydrogen and bicarbonate. Bicarbonate goes in the blood. Hydrogen goes into the lumen of the stomach, and it is pumped into the lumen of the stomach. Remember, gastrin release is stimulated not only by the food in the stomach stretch or increased pH, it also is stimulated by the increased sight of food or smell of food. Mechanical digestion, churning. Not all stomach content is pushed into the duodenum at once, just by small portions, so duodenum can properly digest those small portions. Transport of chyme in the duodenum is controlled by pyloric sphincter, which is located at the junction between the stomach and small intestine. Chyme in the duodenum, in the short term, stimulates gastric secretions, churning and peristalsis. In the long term, inhibits. So this stuff here is about the regulation of digestive activities. So you need to know the cephalic phase. So stimulate gastric digestive activities in the cephalic phase. Sight, smell, taste of food. Parts of CNS that mediate this response, cortex and medulla oblongata. Inhibition of gastric digestive activities, depression and loss of appetite. So depression is through cortex and then medulla, and loss of appetite is hypothalamus through medulla and then to the stomach. Mechanical stimuli in the gastric phase, distension of the stomach, and it goes through the vagus nerve. So vagus nerve transmits those sensory and motor signals between the stomach and medulla. This is called vagovagal reflex. When food stretches the walls of the stomach, signal goes to the medulla, and medulla sends the signal back to increase mechanical digestion. Chemical stimuli, increased pH pretty much, presence of food. Which cells are targeted? Eventually, parietal cells produce more hydrochloric acid. But if step by step, increased pH, more gastrin, more hydrochloric acid. Okay? Chemical changes in the stomach that inhibit gastric digestive activities, lower pH. Okay, pretty much. And again, G cells produce less gastrin, less digestive activities, less hydrochloric acid. What type of nervous stimulation may inhibit sympathetic stimulation? Sympathetic stimulation plays major role in the inhibition of digestive activities. So chemical stimuli stimulate gastric secretions. So in the short term, presence of food in the small intestine increases the gastrin release, and it's a short, a lasting response. But if food stays in the duodenum for a long time, stretch of the duodenum sends the signal to inhibit digestive activities in the stomach. And here, presence of food, pretty much presence of fat and presence of acid in the small intestine and the duodenum, they both inhibit digestive activities in the long term via the release of cholecystokinin and secretin from intestine. Again, when you inhibit digestive activities, it's always through sympathetic. A gastric ulcer can be caused by a tumor, obviously, but most common is Helicobacter pylori. It's the most common cause of gastric ulcer. Loss of mucosal coverage and inflammation. Pathological condition from non-steroidal anti-inflammatory drugs, gastritis, inflammation, the stomach. Duodenum, first part of the small intestine. Small intestine consists of duodenum, jejunum, and ileum. Which organs emptied secretion there? Secretions, liver and pancreas. Gallbladder also empties in the duodenum. And stomach empties in the duodenum. So essentially, stomach, gallbladder, liver, and pancreas all empty in the duodenum. Liver and pancreas empty through the hepatopancreatic ampulla. All duodenal activities are controlled by autonomic nerves. Three structures that increase the surface layer: circular folds, villi and microvilli. Large surface area is important for effective absorption. Columnar epithelium. Microvilli are folds of the membrane on the surface of columnar epithelium. Mucosa-associated lymphoid tissue in lamina propria, as well as blood vessels. Lacteal are blood vessels, mucosa-associated lymphoid tissue, and lacteals are lymphatic vessels that absorb fat. Layer of microvilli and epithelial surface is also called brush border. Importance: it contains enzymes that perform the last breakdown of large biological molecules before the building blocks are being absorbed. Intestinal glands, those are crypts in the epithelium. Cells know the cells, like goblet cells, enteroendocrine cells, Paneth cells. Okay, mucus that's produced by the glands is alkaline to neutralize acid that comes from the stomach. Brunner's gland produces base it's at the very start of the duodenum. Brunner's gland neutralizes acid. What I want you to understand, really, really important. So here, know the function of those cells. Now, this, I'm not going to go over the functions. I have a table with the functions for those enzymes. So you need to know the substrate and the product. Say lactase breaks down lactose into small sugars, okay, monosaccharides. Aminopeptidase, chips of amino acid from one side of the peptide to that extent. So please review them. All these enzymes are located in the brush border. Okay. So secretin is produced by the intestines in response to acid. Secretin increases secretion of bicarbonate from pancreas, and it increases the secretion of bile from the liver. Cholecystokinin is produced by duodenum in response to fats. So it increases the production of pancreatic enzymes and increases and stimulates the release of the bile from gallbladder, release from bile. Okay. So remember, secretin is stimulated by acid, stimulates production of bicarbonate from pancreas and bile from liver. CCK is stimulated by fats, increases the release of enzymes from pancreas and release of bile from the gallbladder. Okay, let's move on. So that I do it in the whiteboard reviews, mechanical digestion in the intestine, segmentation and peristalsis. Liver, that's anatomy. Know the four lobes, what it's covered, it's touched. I'm not going to go over this. Building blocks, liver lobules. Okay, so you've got Kupffer cells, which are immune cells, and hepatocytes. Hepatocytes produce bile and carry out the breakdown of various absorbed nutrients. Portal arteries and veins, they take blood from the intestine into the sinusoid capillaries between the plates of hepatocytes. Blood comes into those vessels from the hepatic artery and hepatic portal vein. Okay, it goes into the sinusoids. In sinusoids, there's an exchange between the blood and hepatocytes. They're really permeable. So that structural feature, they really permeable. After sinusoids, blood goes into the central veins, and central veins eventually carry blood into the hepatic vein. Don't mix up hepatic vein and hepatic portal vein. So to the liver, hepatic artery and hepatic portal vein. From the liver, hepatic vein. And ultimately, the blood from the liver goes into the vena cava, and this blood goes to the heart at the end. Hepatocytes produce bile. Bile ductals carry bile to bile ducts, and at the end, bile goes into the main hepatic duct. And remember, know the anatomy of hepatic ducts, like that main hepatic duct and main cystic duct merge, forming main bile duct. Bile consists of, here's the list. Know their functions. Okay, they're pretty straightforward. Solubilization of lipids, neutralizing acid, solubilization of lipids, result of a breakdown of red blood, of hemoglobin, getting rid of cholesterol, metabolism of steroid hormones, mucin, making things sticky. Bile salts are amphiphilic. They have hydrophilic and hydrophobic ends, so they solubilize the lipids or emulsify the lipids. Okay, both lecithin and bile salts do that. Assemblies of bile salts and lipids are called micelles. Kidneys cannot excrete bilirubin because it cannot pass the membrane, the filtration membrane in the glomerulus. So bilirubin in intestines is converted into urobilinogen, and urobilinogen can be excreted with the urine or with feces. Detoxification, liver breaks down toxins. Kupffer cells protect from pathogens. Carbohydrate metabolism, liver breaks down more complex carbohydrates, stores glycogen, and converts, say, fructose into the glucose. Lipid metabolism, reforming triglycerides, storing triglycerides, and shipping triglycerides out. Major proteins, liver synthesizes proteins essential for clotting and albumin as well. Enterohepatic circulation is connection between the enteric capillary plexus and liver by hepatic portal vein. Bile salts enter the basically they work through positive feedback. They enter the intestines from the gallbladder or liver, and then they are absorbed into the blood from the intestine, go back to the liver through the portal vein, hepatic portal vein, and stimulate their own production. Excess bile is stored and concentrated in the gallbladder. So typical layers in the walls of the gallbladder, bile in the gallbladder gets concentrated. So please know the path from the bile, path for the bile from gallbladder and liver anatomically. Know the duct scheme, as it says. It's pretty much straightforward. So I will ask you, like, for instance, which two ducts form the main bile duct? Well, cystic duct and main hepatic. Gallstones, overconcentrated bile. Cholesterol starts to precipitate together with calcium usually. So risk factors, obesity, lack of movement, blah, 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 all that kinds. Treatment, removal of gallbladder. Pancreas, know the anatomy of the pancreas. It secretes pancreatic juice. The acini secrete pancreatic juice. So bicarbonate neutralizes. So water is the chemical component. Bicarbonate neutralizes the acid, and enzymes break down food. What I really want you to know is the mechanism for production of this chemical. So CO2 interacts with water. It forms carbonic acid. Carbonic acid dissociates into hydrogen and bicarbonate. Bicarbonate is pumped into pancreatic ducts. Hydrogen is absorbed in the water. Main enzymatic components. So here are the enzymes, which I'm not going to go over. You need to know the substrates and the products here. Okay. Gastric and pancreatic enzymes digest them. So proteins are digested by trypsin, chymotrypsin, elastase, carboxypeptidase. That's from pancreas. And dipeptidase and aminopeptidase from brush border. So basically, you really need to know which nutrients are digested in which part of alimentary tract. For instance, proteins are digested in the stomach and small intestine, or carbohydrates are digested in the mouth and the small intestine, and so on and so forth. Mechanism of amino acid transport. So it's really straightforward. So on the basal side of intestinal epithelium, you have sodium-potassium pump, which creates sodium gradient across the apical side. The sodium gradient drives secondary active transport. So, for instance, amino acids are transported by secondary active transport on the apical side. It's active transport, and then they diffuse via basal side. They diffuse in the blood. Same is true for carbohydrates. So know which enzymes break down oligosaccharides across the alimentary canal. So monosaccharides, active, secondary active transport on the apical side and diffusion at the basal side. Essential driving force is sodium-potassium pump that creates sodium gradient across the apical side. Sodium-potassium pump is located at the basal side. Lipids, again, know which enzymes digest them. So they're broken down into diglycerides, so monoglycerides and fatty acids. Fatty acids and monoglycerides are transported into epithelial cells by micelles. They do not require transmembrane proteins because they are fat soluble. In epithelial cells, they recombine into triglycerides, and triglycerides are packaged into so-called kilomicrons or chylomicrons, and chylomicrons, packaging of triglycerides with proteins, enter the, so they need to have proteins to be more or less soluble into the lymph where chylomicrons go, and they end up eventually in the blood and then liver. So it's a review. A lot of it is a review. Okay, parts of large intestine, know the anatomy, like ileocecal valve, ileum, the cecum, ileocecal valve, cecum, ascending colon, transverse colon, descending colon, sigmoid colon, rectum, anus. So just know it. Absorptive, propulsive, compaction, churning, forming feces. Absorptive, mostly water in some ions. Cecum and appendix, most likely storage of microbiome. Function of rectum, expel feces. Commensal bacteria, the immune system. I'm not going to ask you much about this. Epithelial lining in the large intestine. Columnar. In the anal canal, columnar, simple columnar changes to stratified squamous. Columns and sinuses in the anus produce mucus. Various mesenteric structures support large and small intestine. So, okay, parts of the colon, epithelium, columnar, already got this. Intestinal glands secrete mucus, basically. Water and some ions are absorbed in the colon. Mold protects from infections. Taenia coli, responsible for generation of so-called mass movements. Haustra are individual anatomical pockets in the large intestine. They participate in segmentation. Mass movements, powerful contractions of the large intestine that usually result in bowel movements, in defecation. Stimulus, main stimulus, consumption of food and release of gastrin. Slow waves, so pacemaker cells generate slow waves. Peristaltic reflex, stretch. At local plexus, signals are integrated, and then smooth muscles behind the bolus contract in front of the bolus, relax. So like contraction, like acetylcholine stimulates contraction of the smooth muscle, and acetyl and nitric oxide stimulates the relaxation of downstream smooth muscle. So no, there are actually 4 neurotransmitters. You can find them in the notes. Defecation, stretch of rectum, signal goes to the local plexuses and to the brain, and then response is relaxation of the internal anal sphincter, that's involuntary, and then brain gives the signal when it is ready to relax external voluntary sphincter, which is voluntary. So basically, internal is autonomic, external sphincter is somatic, under control. Okay. So external is voluntary, internal is involuntary. Now, parasympathetic stimulation relaxes sphincters, and sympathetic stimulation contracts. Infants cannot control external sphincter. Vomiting can be caused by blockage of the intestines or, irritation of the stomach. Vomiting reflex, let's say irritation of the stomach, signal goes to the medulla oblongata, which controls vomiting reflex. Medulla in the vomiting center releases chemicals, and they stimulate reverse peristalsis. Electrolytes are absorbed in the small and large intestine. Sodium is absorbed, as we discussed, via sodium-potassium pump at the basal side, which creates the gradient of sodium across the apical membrane. Water is absorbed by osmosis, okay, and it follows the influx of sodium. Potassium is absorbed usually by the transport between the cells, because when water is absorbed, potassium concentration in the intestine rises. Calcium is absorbed when needed, and parathyroid hormone controls its absorption. Iron is absorbed only when needed, so there's no point of, getting iron pills. Celiac disease is the disease, immune response to gluten, autoimmune response to gluten, which results in a mass of T cells migrating to large intestine and just causing inflammation and disruption of proper absorptive membrane. So that's anatomy. I'm not going to go over anatomy, obviously. That is really useful, primary, secondary, and main functions. Description I'm not going to ask you to describe it. It's useful, like fill all this out. That's for cephalic, gastric, and intestinal phase of the digestion in the stomach. Nutrients that are digested in different parts of the alimentary tract. Generation of the acid and bicarbonate in acinar cells and parietal cells. Function of the enzymes, sorry, the alignment was off. Mechanisms of absorption that you can draw using the review videos, they're really useful. And we did not talk about this, so you don't have to worry about that part. So I hope it all makes sense. I'm going to upload it and you will see it.